Welcome to Decoding Healthcare Research. I'm your host, Efrain Riveros, Dr. E. Efrain. Today, we are going to talk about the gut permeability in particular, and very interesting experiments and additions to the science that have been made by an interesting group in Sweden. We have a researcher here with us who is an expert in this topic. We have Dr. Maria Fernanda Roca Rubio. She's a researcher of the Nutrition Gut-Brain Interactions Research Center, School of Medicine, Medical Sciences in Örebro University in Sweden. Welcome, Dr. Roca.
Thank you very much. I'm very happy to be here.
Excellent. Thank you for being here with us to share your knowledge with us. So we are going to talk about a couple of papers that her team published. One of them was published in Scientific Reports of Nature Portfolio in 2021. The title of the paper is Sauna Dehydration as a New Physiological Challenge Model for Intestinal Barrier Function. And we will also talk about a paper that was published in the PLoS One journal in 2021. The title of the paper is Short Intense Psychological Stress Induced by Skydiving Does Not Impair Intestinal Barrier Function. So the first question for Dr. Roca is why intestinal permeability? And tell us about your line of research.
So my line of research during my PhD thesis, these are all the papers from my PhD thesis, and was intestinal barrier function. And this is because the intestinal barrier is the largest barrier between the host, so the person, and the external environment. And therefore, like it protects the host, so us, from different pathogens, or substances that may be harmful. And it's therefore very important as it is formed by many cells that are lined up together. And if the barrier is disturbed, then such like pathogens or like harmful substances can cross, and then it can cause an infection.
And this is interesting, and it's becoming actually very central, as we have been in other episodes of the podcast talking about the role of inflammation in the gut and systemic inflammation. So everything is interconnected, and definitely the intestinal barrier is playing a significant role in many disease processes. And as I understand, you guys have been trying to develop reliable models to investigate the role of different strategies aimed at intervening on the intestinal barrier. So let's start with this first one related to sauna. Why did you guys think that sauna could be an interesting model? How does it affect the intestinal permeability barrier?
So this model comes from a previous experiment, previous research that has shown that high intensity exercise can increase intestinal permeability. So but it's not exactly known what of exercise may be the cause, because it's usually, it could be like the bouncing movements of exercise that are affecting the gut. It could be like the dehydration that is caused by strenuous exercise. It could be also the psychological stress, or it could be many things. So then we thought, okay, let's take two out of all of these factors and see if those work. And then we decide on the sauna model, because then we have increased body temperature, and we also have dehydration. So that's where the model comes from. And then, yeah, we use the sauna to try to increase intestinal, like to see if it will increase intestinal permeability in healthy subjects. And well, it did. I can tell you about how we did it, or
Perfect. No, that's. Let's go a little bit deeper into the methods that you guys used. Obviously, this is the exposure or the intervention. In this case, is the sauna itself. But I assume that in order to see the effects on the intestinal permeability barrier, you guys had to make some measures systemically, like probably blood samples. What did you measure to try to assess the effect of sauna on permeability?
yeah, in this paper we use a model that is test that is called the multi-sugar intestinal permeability test, and it consists on the intake of 5 different sugars that are different in size, and therefore they are going to be absorbed in different parts of the gut. So first we have sugar, sucrose, that is regular sugar, and it's absorbed in the gastroduodenal part of the gut. And then we have the lactulose and rhamnose. They are 2 sugars, and are like the ratio between them is calculated. They are absorbed in the small intestine. And then the last 2 sugars are sucralose and erythritol. They are absorbed in the colon, and they are used as a marker of colonic barrier. All the sugars are the participant takes the sugars in dissolved in water, and then we are recovering it in urine. So the participants have to collect urine during 24 hours into different containers. One of 5 hours, where we are going to measure gastroduodenal and small intestinal permeability, and the other one of 5 to 24 hours, where we are going to find sucralose and erythritol. And that's the markers that we use for permeability. But we also use blood samples to assess intestinal fatty acid binding protein, which is a small protein that is secreted, when it sits on the top of the villi of the enterocytes, and then when there is some disturbed, and then it's the first protein that is gone into the systemic circulation, and then we find it in the blood. We also find LBP, which is lipopolysaccharide binding protein, which binds to LPS, which is it's a part of the bacteria cell wall. And we also measure some cytokines for inflammation.
Excellent. So now we have the exposure, we have what you measured. So let's talk about the intervention first. So how can you describe the protocol? For how long were people exposed to the high temperatures of sauna? And when did you consider that dehydration was happening?
Yeah, we decided that we were going to aim for 3% dehydration in all of our participants. So for this to happen, they arrive in the morning, they had to be hydrated. They were normally hydrated, and then we asked them to go into the sauna. And they went into the sauna for 15 minutes. They were in the sauna, and then they were out of the sauna for 10 minutes. In these 10 minutes, we take body weight. We weight them. And then they went again into the sauna until they had lost 1.5% of their body weight due to dehydration. And then it's when they drank the multi-sugar solution. And then they continue the in and out protocol until they had lost 3% of their body weight due to dehydration. And then is when we took the saliva. We took saliva samples, we took blood samples, and the urine we started collected from the moment they drank the sugar solution, and they had to urinate.
So how long was the average for people to be in the sauna?
Yeah, not just in the sauna, but it took around 3 hours from them.
3 hours for the whole protocol, in and out.
Yeah.
Excellent. So let's move on to the findings. So what did you find? And this is exciting because all the things that you measure, not only at the local level, but the systemic inflammatory level is interesting. So tell us about what you guys found.
So we found that there was indeed an increase in intestinal permeability, in a small intestinal permeability compared to the control condition. We also found that there was an increase in small intestinal permeability compared to the control condition, and it was really interesting that there was not an increase in colonic permeability. So we think, well, we don't think that we show that this is a protocol that actually works to increase intestinal permeability. And we also found that there was not an increase of IFAP, which is a marker of enterocyte damage. So that's like it increased permeability without damaging the enterocytes. We found that to be very interesting. There was also, there was also an increase in LVP compared to the control condition. So it might be that there was an increase in so that some bacteria passed, but LVP is also a marker of immune activation. So it could be that just the heat triggered this immune activation. But we saw an increase in this marker, and we also saw an increase in some cytokines, such as interleukin-6 and interleukin-8. And interleukin-6 has also been shown to be increased in exercise conditions. So it could be related to the increase of LVP. And interleukin-8, it was also increased. And I think this one has also been shown to be increased in cells when they are dehydrated.
Yeah, and it's interesting to see the similarities between exercise and sauna, as you point out. And I think also that this protocol gives us, sheds light on not only the potential for sauna as an inductor of gut permeability increase, but at the same time it shed some light on mechanistic ideas, because what you mentioned, that it doesn't really cause epithelial damage or gut barrier physical damage. What we see is the increase in permeability, but at the same time preserving the function is really interesting, and it may be in line with the beneficial effects of exercise and sauna. That's pretty interesting. So I saw that you guys measured cortisol in saliva. Why did you guys do that? What is the goal of measuring it?
Yeah, we measure cortisol as a marker of psychological stress. And we saw that there is a peak of the cortisol right after the dehydration protocol. So when they went out, and I mean, it could totally be for psychological stress because they had been there and then they are about to draw blood, and that can cause some psychological stress. But I have to say that back then they were actually very dehydrated. So it could also be that the concentration was higher in the amount of saliva. So like in that moment.
And I see that, I'm sorry, go ahead.
No, just tell me.
I see that you guys run a series of correlations precisely between cortisol and intestinal permeability, and also cytokines and permeability. What was the idea of running all these correlations?
Yeah, well, we wanted the different correlations. For example, the one of cortisol with the different markers is because psychological stress has been shown to be linked to an increase in intestinal permeability. So we wanted to know, we wanted to rule out that the increase of intestinal permeability was not because of the psychological stress, that it was because of the sauna dehydration. And that's why we did all these correlations. And then the correlations between the markers of all the markers against the intestinal permeability, like measured by the sugar test. And those we did because the sugar test, although it is a very good method and it's not really invasive, it's very strenuous for the participants, as they have to be collecting their urine for 24 hours, and it's not so easy for them. Like, it is easy, but it's annoying for them. So we wanted to find a marker that could substitute it, and a blood marker will be easier to it will be more invasive, but it will be more it will be faster. And
faster.
yeah. So, and then about the temperature, we wanted to know if the temperature by itself would have was already related with an increase of any of these markers. So yeah.
I feel that one of the aspects that was really intriguing to me, and I want to dig deeper into that, is that you mentioned that the lipopolysaccharide binding protein, which actually you guys measured, that one is produced both by the intestine and the liver. This particular molecule is the or completes the interaction with the immune system through receptors probably in the T cell. Can you tell us a little bit more about the role of this protein and why is it important that you measured it?
LVP, yeah. So when there's an infection, like when there is some bacteria passing, the bacteria's cell wall has it's like the gram-negative bacteria, I think. Gram-negative bacteria cell wall is composed, it's formed by LPS, that is lipopolysaccharide, and this is like the agent that will start like in the toxin, like the reaction that is like the bad guy, and then it will pass, like when passes the barrier, and some different things will gonna try to trap it, and will gonna try to stop this, like to make an infection. So when it will be attached, it will be LVP, will be attached to LPS, and different LVPs will attach to another molecule that is called soluble CD14. And together with this, it starts the chain to, like the immune chain to stop the infection. And it like then SCD14 binds to another receptor that is, I think, TLR4. And then, yeah, it starts the immune cascade. However, LVP not only happens when there is endotoxin translocation, but it is also activated when there is systemic inflammation. So it doesn't necessarily have to come from the gut. Like it doesn't have to be just like something past the barrier. You can have some infection and it's a liver protein. So it's a CRP, it will also be segregated. And when I was writing this, I did knew it, but I didn't understand it like so much. So like after I was like, oh yeah, it's like it maybe is because interleukin-6 is activated and the exercise, it's like all triggers. So maybe that's why it's trigger LVP, but it could be that something passed.
So it's very interesting. So now moving to the intestinal permeability part, what are the postulated mechanisms by which you think sauna is associated with increasing intestinal permeability? What might be happening at the cellular level?
Sorry, once again.
Yeah, what I'm saying is that definitely you guys demonstrated that sauna increases intestinal permeability. So what are the postulated mechanisms? What is happening there? Because you guys demonstrated that there is no cellular damage per se. So how does the increased permeability happen?
Yeah, so it could be by different things. It could be tight junction damage or tight junction breakage, or it could also be like shrinkage from the enterocytes. So like as they shrink, then maybe there is more space for like more permeability between the cells. Mm-hmm. More, so it could be that, or like yeah. I think those are the.
So it's mostly shrinkage. Yeah. You guys think that's rather interesting. So let's talk about the limitations of the study. What do you think could decrease a little bit the validity of the study? Are there any important limitations?
I mean, it's the biggest limitation. I think it could be the trial per se. So it is kind of strenuous for the participant, although it's easy and it's not expensive, the protocol. It could be that, like, yeah, being 3 hours sitting on a sauna, like in and out, it's not so comfortable. And it was like that. Another of the limitation is that, I mean, if we will have a biomarker that it can be easily available in blood, like you can do just an ELISA test and have the results. But here, like with the multi-sugar test, you need special equipment. So you need an HPLC liquid chromatography mass spectrometer to assess it, and you have to have it calibrated. So like, although it's not such a complicated experiment, it's still not available in all the labs. Yeah, I think those would be like the biggest limitations.
Yeah, but overall, it's a very interesting paper, and I'm sure it opens a window for testing different strategies that could affect or restore intestinal permeability, and I find a lot of value in this. I also saw in your other paper that using the short stress of skydiving was evaluated, and the exposure obviously is different here, but the measures are rather similar. So can you tell us a little bit about that protocol and what you found?
Yeah, so in the skydiving experiment, we thought, because it has been shown that in the that psychological stress can increase intestinal permeability. We thought, okay, like what could be like a very intense acute stressor that could increase intestinal permeability, because this has been just shown with a speak test, and it was a master defense test, and then we thought like maybe it's not acute enough because people is already stressed for a long time, and we thought like an acute stressor could be skydiving. So yeah, we decided skydiving as an acute stressor, and for this the participants had to be willing to skydive, and then we went to the skydiving club and we bring them up there. And yeah, the participants jumped, and we took saliva samples before, when they woke up, before they were in different time points, but it was basically when they woke up, 2 hours before they skydive, and then right before they go into the plane, and exactly within 5 minutes when they land. So we were there waiting for them to land, and we gave them the saliva test, and then in the skydiving club, also within 5 minutes, we took them to the lab that we have put there, and we took blood samples we collected, and they also took the multi-sugar test right before they went into the plane to measure intestinal permeability. And yeah, in this study we saw an increase in cortisol, so there was a clear peak in cortisol after they land, but we thought they will be stressed, much more stressed before, and they are not really that stressed before they do the jump. So maybe it was too acute, so we did not saw an increase in intestinal permeability.
Yeah, probably to study that the exposure to stress has to be long lasting, and that's true. Yeah, and it's interesting when you combine both reports to see how you guys are being able to isolate the different effects, say, of exercise on the intestinal permeability, and that's an added value of the papers, and I appreciate it. So I think this way we are coming to a close, saying thank you. Thank you, Dr. Roca, for sharing your knowledge with us, your wisdom, and these experiments are very not only intriguing, but they basically, as I said before, open windows for new lines of research trying to investigate the interventions on gut permeability. As we know, more and more publications are coming out relating intestinal permeability not only to local problems like IBS or even colon cancer, but to systemic diseases. And inflammation is central there, and also the intestinal permeability. So thank you, Dr. Roca, for being with us today.
Thank you. Thank you very much.
It was great. And for our audience, the links to the papers you will find in the comments, in the description of the video. And don't forget to leave us the feedback in the comment section. See you in the next episode.